Welcome to the second part of overview of immunology. Previously we discussed the history and discoveries of immunology as well as the definition and types of immunity. We also discussed innate immunity and its barriers in details. Link is in the description if you wanna see part 1 of this video. In this video, we are gonna discuss about the discovery of humoral and cell-mediated immunity, non-specific defense barriers of human body, endocytic and phagocytic barriers, and barriers of inflammatory response. The concept of humoral immunity was developed in the late 19th century by Hans Buchner, Emil von Behring, and Paul Ehrlich. Their work was based on the study of blood serum and its antibacterial properties. Hans Buchner is credited with the development of the humoral theory. In 1890, Buchner described substances that exist in the blood serum and other bodily fluids and are capable of killing microorganisms. In 1890, von Behring and Kitasato Shibasaba discovered that cell-free filtrates from diphtheria and tetanus could cause disease. They also discovered that immunized serum could neutralize the toxins and transfer immunity to others. In 1897, Paul Ehrlich showed that antibodies form against the plant toxins ricin and abrin, and proposed that these antibodies are responsible for immunity. In the early 1900s, researchers characterized the active immune component in blood serum. This soluble component could neutralize or precipitate toxins and could agglutinate, or we can say clump, bacteria. This component was named for the activity it exhibited, that is antitoxin, precipitin, and agglutinin, respectively. In the 1930s, Elvin Cabot found that a fraction of serum first called gamma globulin, which is now named as immunoglobulin, was shown to be responsible for all these activities. The soluble active molecules in the immunoglobulin fraction of serum are now commonly referred to as antibodies. Because these antibodies were contained in body fluids also, the immunologic events they participated in was called humoral immunity. Passive immunity is a type of immunity that occurs when a person is given antibodies rather than making them through his or her own immune system. For example, passive immunity occurs when a baby receives a mother's antibodies through the placenta or breast milk. It can also occur when a person receives an injection of antibodies to protect against the such as snake venom. Passive immunity is used as a preventive to boost the immune potential of those with compromised immunity or who are at high risk to acquire infection. Passive immunity provides immediate protection but only lasts a few weeks or months. Active immunity is a long-lasting defense mechanism that occurs when your body's immune system produces antibodies to fight off a disease. It can be acquired naturally through infection or artificially through vaccination. Active immunization stimulates the immune system to produce antibodies against a particular infectious agent. An individual who recovers from a first case of the measles is immune to further infection. Cell-mediated immunity, or CMI, is an adaptive immune response that helps the body fight off foreign substances like viruses, bacteria, and poisons. CMI doesn't involve antibodies, but instead activates other immune cells and releases cytokines. Cell-mediated immunity is directed primarily microbes, parasites and microbes that infect non-phagocytic cells. It is most effective in destroying virus-infected cells, intracellular bacteria, and cancers. It also plays a major role in delayed transplant rejection. Here's how CMI works. First, phagocytic cells, like macrophage cells, recognize a foreign particle. Then the phagocytic cell engulfs and digests the particle, but some small molecules called antigens remain intact. After that, the phagocytic cell presents the antigens on its surface with the help of major histocompatible complex, also known as MHC proteins. Lastly, the M complex activates cells of the adaptive immune system, which includes CMI. CMI is one of the body's adaptive immune responses, which are tailored to specific pathogens. Adaptive immunity can last for weeks, months, or even a person's entire life. Ever wondered how foreign substances are recognized by immune system? They do this by recognizing substances called antigens on the surface of the microbe, or in the chemicals they produce, which mark the microbe or toxin as being foreign. The antibodies then mark these antigens for destruction. There are many cells, proteins involved in this attack. Antigens are proteins on the surface of cells, viruses, fungi, or bacteria. Non-living substances like toxins, chemicals, drugs, and foreign particles can also be antigens. Every cell or substance has its own unique antigens. First, the immune antibodies that match specific antigens. Then antibodies attach to the antigens, making them harmless. These antibodies also activate other immune system cells to help fight the antigens. The process of pathogen recognition involves an interaction between the foreign organism and a recognition molecule, or molecules, expressed by host cells. It can be through membrane-bound receptors, soluble receptors, or secreted recognition molecules. Ligands bind intracellularly or extracellularly and destroy pathogens. Ligands for these recognition molecules can include whole pathogens, antigenic fragments of pathogens, or products secreted by these foreign organisms. In case of response to intracellular interaction, recognition of virus-infected cell take place by cytotoxic T lymphocytes. The recognition molecules are present inside the cells. These intracellular receptors bind to viral proteins present in the cytosol and initiate an early warning system and destroy the pathogen. In case of response to extracellular interaction, extracellular pathogens are recognized by the receptors present on cell surface or soluble recognition molecules that probe the extracellular spaces of the body. In this case, B cells and the antibodies they produce as a part of humoral immunity play major roles. The theory of immune specificity is the idea that the adaptive immune system can distinguish between different pathogens. This ability is a fundamental property of adaptive immunity. Two major theories were proposed to account for this specificity: the selective theory and the instructional theory. The concept of the selective theory was given by Paul Ehrlich in 1900. He explained the origin of serum antibody. Ehrlich proposed that cells in the blood expressed a variety of receptors, which he called side chain receptors, that could bind to infectious agents like the fit between a lock and key and inactivate them. This enters the cell to produce and release more receptors with the same specificity. The cells were pluripotent, expressing a number of different receptors, each of which could be individually selected. Each cell makes many copies of just one membrane-bound receptor of one specificity. An army of cells, each with a different antigen specificity required. Then the selected B cell can be triggered to proliferate and to secrete many copies of these receptors in soluble form, which are now called antibodies once it has been selected by antigen binding. The instructional theories were formally disproved in the 1960s. Now the information was emerging about the structure of protein, RNA, and DNA give the new insights of how an individual could make antibodies against almost anything. A dysfunctional immune response occurs when the immune system is overactive or underactive, or when it attacks the body's own tissues. This can affect the body's ability to fight off infections and vaccines. Hypersensitivity is an immune system abnormality that causes an extreme response to a substance, such as an allergen or drug. This response can lead to undesirable effects, such as allergies and autoimmune diseases. If the person have an autoimmune disease, your immune system attacks the healthy cells of your organs and tissues by mistake. There are more than 80 types of autoimmune diseases. They can affect almost any part of your body. Alopecia areata is an autoimmune disease of the skin that causes hair loss. Immunodeficiency is a condition where the immune system is unable to fight off infections and cancer. It's also known as immunocompromisation. Immunologic memory is the ability of the immune system to respond with greater efficiency during a second exposure to the same pathogen. Primary response is a first encounter with foreign antigen. Lymphocytes will be used to eradicate the pathogen are clonally selected to resolve the infection. Whereas secondary response is all subsequent same antigen or pathogen. Memory B and T cells play important role in secondary immune response. And it is also important in vaccination. Here are some important features of immune system. The microenvironment in which the immune response begins to emerge can also influence the outcome. The same pathogen may be treated differently depending on the context in which it is encountered. Some areas of the body, such as the central nervous system, are virtually off-limits for the immune system. Response could do more damage than the pathogen. Some foreign compounds that enter via the digestive tract, including the commensal microbes that help us digest food, are tolerated by the immune system. However, when these same foreigners enter the bloodstream, they are typically treated much more aggressively.